I am really humbled to be in the UK for the first time this week. I've had the pleasure of speaking to the Chalfont Beekeeper Society, the Harrow Beekeepers, and now at the National Honey Show. And it just brings me immense pleasure to see so many of you new faces to me, and in this wonderful country that has given us a lot to talk about for the next few months when we go home to the US. And I would like to thank Simon for inviting me to this National Honey Show. It's been already a tremendous experience. I conducted my PhD with Dr. Tom Seeley. A lot of you have heard of him and probably have read Honeybee Democracy, so it always brings me pleasure to reminisce about my time when I was working with Tom. And when I was there, I was doing work on the biology of swarming, which is going to be a talk that I'll give tomorrow. So tomorrow's talk is going to be more, it's going to contain a lot more data and really cool stuff and videos about the work that we did with Tom when I was getting my PhD at Cornell University. This talk is going to be more just plain biology. So for some of you who are advanced beekeepers, a lot of this information might be repetitive or not that exciting. But as Simon said, it is, to me, for some strange reason, it's fun to talk about drone biology and how you can assess drone reproductive quality and sexual maturity. So why talk about the biology of mating? Well, even for not just the beginner, but any beekeeper, it is really, really important to know exactly when mating is happening, what is happening during the mating season, so that we can prevent and or prepare, be it to have sufficient numbers of drones around near our mating yards, or what is happening between in the birds and the bees of bees to prevent swarming and or mating, if that's the outcome we're trying to prevent. So the outline for today's talk is going to be just a quick, very quick and dirty 101 differences in the castes of the colony, just to put all the beginners up to speed and remind everyone about the different castes within the colony. Then what conditions induce queen rearing in a colony, because typically there's only one queen in a colony, but in 3 specific conditions there are multiple queens or there's polygyny in the colony, and so we'll talk about those. And then we'll dwell into the biology of the queen and a lot of the physiology that is important to remember when we're talking about mating, and the same for drones. And at the end, we're going to add A plus B to get the mating biology, and we'll talk about the biology of when mating occurs, the DCAs, the mating signs, etc. As I said, right now for this talk, we're just going to do basic biology between drones and queens. And so we're going to talk about the conditions that are inside a colony that's preparing to rear queens and drones for mating, which is very important for beekeepers to know. So exactly when does mating occur, and how do we take advantage as beekeepers of the bees' biology to, for the purpose of rearing queens and drones and for preventing swarming, if that's what we want to do. So let's remember that there's 3 castes in the colony. The female workers are facultatively sterile, but in certain circumstances, especially when you have a queenless colony, certain lines of workers will develop some of their ovaries and lay unfertilized eggs that will turn into drones. But typically they're sterile, they are smaller in size, they have stinger for colony defense, and they have pollen baskets, the corbicula for pollen collection, have several glands that produce the brood food, all of the defense secretions, and of course the wax that makes all the comb. The drones, we like to call them flying sperm cells, because really their sole purpose is to mate and pass on their genetic material to next generations. They have really large eyes, and that aids them in locating queens up in the drone congregation areas for mating. They don't have a stinger, they don't have corbiculae, and also they don't have glands for performing pretty much no task besides mating. So they're really expensive to produce, and they're only produced during the mating season if the right conditions are met inside a colony that is quite strong for doing so. Then we have the queen, Your Majesty, who is the egg-laying machine, depository of all the genetic material in the colony, and she has no corbicula, no pollen baskets, because most of her glands will have to do with secreting pheromones that will maintain colony organization, and we'll go into those further. So when a queen lays an egg, we know that she has the ability to decide based a lot on physical cues, what kind of cell she's inspecting, and depending on the width and size of that cell, she makes the decision of whether or not to fertilize the egg she's about to lay. So if she does not fertilize it, that individual has only one set of chromosomes that comes from her, 16 chromosomes in total, and the individual develops into a male drone. If that egg is fertilized, then it will have two sets of chromosomes, one from her and one from one of her many partners. And then the worker later on will determine whether to raise that larva into a worker or a queen. So those are the fertilized females in the colony, are either the worker or the queen, based solely on nutrition. So when queens are being reared, what is happening inside a hive? Queens are reared in three conditions in a colony. The most natural and abundant time for queen rearing is swarming. Colony fissioning, which I will talk about tomorrow. Very cool stuff happening inside a colony that's getting ready to swarm. So during the reproductive season, a colony that's large enough will decide to split into two units. The swarm colony will depart with the mother queen and about 3/4 of the worker population, whereas back in the parental nest, the colony will have started a queen rearing process that can have upwards of a dozen or more queen cells at different developmental stages, that one of which will develop into the queen that inherits that colony. And that's how colonies basically increase their fitness, is by splitting. If they're lucky enough to split that in that year, then their fitness goes up. Then the other two scenarios of queen rearing are emergency queen rearing. So typically, I forgot to mention, when you're having a swarming condition, you typically find the cells at the bottom of the frames where there's more space, or inside in a tree cavity for feral colonies, then you would find the cells at the edges of the comb, because there's a lot more space and they're vertically oriented compared to the horizontally oriented drone and worker cells. But in emergency queen rearing, which happens when there is no queen, the colony goes suddenly queenless, because the queen either got predated upon if she was maybe out mating, if she was a virgin, or if she died because she was ill or something like that. Then the workers will go into this emergency mode. They will try to rear a queen from the middle of the brood nest, where worker larvae will be raised. And then the workers will elongate and change the orientation of that worker cell into the peanut-shaped queen cell that we know about, but typically is in the middle of the brood frame, and you have fewer emergency queen rearing cells. So a good indication for a beginner beekeeper of whether your colony, not just seasoned, but whether your colony is about to swarm or it has a different kind of queen rearing, is where you locate those cells. So if they're in the middle of the brood nest, they could be doing emergency queen rearing or supersedure, which is a very problematic issue in the United States right now. So supersedure is the sudden replacement of the mother queen with a new queen by the workers, and we still don't understand why workers are deciding to replace the queen with a new one, but we know that they will do so also kind of in the same manner. They will find some fertilized larvae in the middle of the brood nest and decide to raise her as a new queen, and they will not just do one, they will put more eggs in a couple more baskets and rear a couple more, and one of those queens will eventually become the replacement queen of that old queen. So the difference between these two is that in supersedure, the mother queen is still around. And in some cases, because we all know as beekeepers that bees are not reading the honeybee biology book, you will find failed supersedure attempts whereby the workers will have tried to raise a new queen to replace the mom, and then suddenly they choose that after even the virgin queen has emerged, they decide not to kill the mom. And sometimes you have these situations of a polygynous colony with a couple of laying queens side by side, and they seem to be allowing each other to do so. So that's a very interesting phenomenon that not many studies have been done on because it doesn't happen very often. But let's just focus on swarming and what conditions are happening inside a colony that's getting ready to swarm, because right upon swarming, right, a few days after swarming is when mating occurs, right? The virgins will try to mate with drones. So this is just a diagram that you can find in the very popular Winston honeybee biology book of the time when swarming occurs. So here in this x-axis, we have the months of the year, and then here we have a relative amount of colony population size. So not surprisingly, the colony size is the lowest coming after the winter because, of course, they've depleted resources and you have the winter bees. But as soon as you have a little bit of resources coming in and the temperature goes above a threshold of, say, in Celsius, what would that be? So 55 Fahrenheit. 10, 15 degrees, you will start seeing some bee activity. And the especially egg laying, so you have a rush of an increased population of young workers that are starting to come out, and the population explodes. And at least in the temperate northeastern region of the United States, which is probably a little similar in many ways to around here, you see most of the swarming occurring in May, June, and a little bit in the month of July, when the colony population reaches a threshold level whereby it has to split into two units because the cavity that it's in cannot contain any more brood or adults, etc., and it splits. So this here is a blow up of what we're seeing. That up here is what we have down here. So day 0 is the day when the colony swarms, when it decides to split. This is just relative numbers, so we know what's going on inside. About 10 days or a week or so before splitting or swarming, the colony will have started the queen rearing process right here, because it will not split without having at least one cell that is sealed, so that there's some insurance policy that there will be at least one queen that will inherit the nest. Then the colony splits, and a few days later, maybe 10 or 20 days later, you might have after swarming going on. And so the after swarm is headed by a daughter queen, not by the mom, because the mom already left. And so the after swarms might be headed by still virgin queens that will leave with a very small proportion of the colony workers that remain behind when this first swarming happened. And then about 20 days later is when egg laying, right here, when egg laying starts or resumes once those virgin queens or the virgin queen goes out and mates properly. So there are several stimuli, physical stimuli, that are telling a colony that it's time to start the queen rearing process because swarming is about to happen. So imagine that this box is the confined space of either a tree cavity or even your beehives when you're keeping bees as a beekeeper. So inside we have colony size constraints. There's only so much area and volume that can be occupied by the bees. We as beekeepers try to increase that size by putting on maybe a couple of more boxes on top to aid the bees in having more space, but in a tree cavity it's difficult for the bees to find any more space. They only have so much room to grow. So limited cavity volume and area. There is congestion in the brood nest. So the queen is running out of room to lay eggs. There are no available cells for her to lay eggs. Most of them are occupied by brood workers and drones. They are rearing drones that are, again, as I said, seasonal. So there's really no room for growth. There's also no room to put away more resources, especially when you have this increase in resource abundance of nectar and pollen that is kind of a positive feedback of putting in resources to allow the colony to produce more brood, etc., and kind of outgrowing itself. Then you have that first batch of workers that's coming out from the first thousands of workers that were laid a couple of weeks prior. So we have a low average age of workers with a high population of very young workers, which are typically the workers that leave with a swarm, with the queen, are the kind of middle-aged workers that are old enough that they have already started their foraging career, so they know what's out there, they know the landmarks, they know areas where they could potentially live, but they're young enough that once they swarm, they will still have a couple of weeks of lifetime that they can dedicate to the buildup of the comb that will be used for that swarm colony in its new home. This congestion basically will lead to a reduced transmission of queen pheromones, and I will talk about those in detail. So there's very low, there's a reduction in the transmission of QMP and other pheromones because there's no room for the workers that pick up those molecules from the queen to pass on to the workers around them. So typically, there are very low titers of queen pheromones in the periphery of a colony that is getting ready to swarm. That indicates to those bees that it's time to start constructing the cups, queen cups, and the queen herself will start laying eggs in those cups, and the queen rearing begins, and then swarming can occur. So there are three very important scenarios that we as beekeepers need to keep being aware of during the swarming season, is that or for queen rearing, if we are in the business of rearing queens, and we can talk about queen rearing outside of these lectures if you want, because I did a lot of queen rearing training for beekeepers in the eastern United States. We still do that a lot in Texas. So you have a lot of resource abundance, including pollen as a protein source that will then be turned into brood food. You have a population of young workers, and if you're doing any queen rearing, then those workers will be nurse bees, the youngest workers that will take care of those queen cells that are being reared. And then you have either a reduced transmission of QMP, or for the purpose of queen rearing, for instance, you don't want a queen. You want a queenless scenario so that there is no queen pheromones, because amongst other things, queen pheromones inhibit queen rearing, and we'll talk about that. So very important things for queen rearing is resource abundance, high population of young workers, and no queen. Okay, so let's talk about the queen in detail. Typically, but this can change depending on weather or nutrition, the queen will take 16 days from egg to emergence. So all 3 casts will stay in the egg stage for 3 days, and then they will have typically plus or minus some hours, but 5 days as a larval stage, because honeybees, as you know, are holometabolists. That's the entomological word for saying that they have complete metamorphosis. So they have an egg stage, a larval stage, a pupal stage, and an adult stage. The larval stage is the stage of growth, and they basically eat their body weight several times over and increase in size exponentially over the 5 days of larval growth. What differs between the 3 casts is how long they will be in the pupal stage. For queens, they will take basically 16 days from egg to emergence. So on day 9, the larva is capped, and on day 16, the queen emerges. As I mentioned earlier, fertilized eggs laid by a queen are bipotent. They have the ability, or they have the flexibility or plasticity to become either workers or queens based on nutrition. So there isn't a queen destined egg per se. Almost any fertilized larva, depending on how young it is, will be able to be raised as a queen either by workers or by us. But a queen will develop from a fertilized larva that gets fed that really proteinaceous royal jelly. So you typically find these really huge pools of royal jelly that almost look like too much food. They look like they are being overfed sometimes if your colony is nice and hefty, which is really good to see. And people that rear queens, they will go into colonies that have started any kind of natural queen rearing process, and they scoop out that royal jelly out of their colonies and they use it for their own queen rearing purposes, which is a very good way of increasing nutrition during artificial queen rearing by beekeepers. In beekeeping, we typically say that your colony is either queenless or queen-right. And a queen-right colony is a colony that has a functioning queen, and it functions as this very cohesive unit, mostly because of the queen-derived pheromones, mostly the QMP or queen mandibular pheromone, but there are other very important pheromones that she releases from several glands that are transmitted about the colony through worker-queen contact. So very important female in a colony that handles or mediates decentralized control of colony organization. Because workers are not really coming, not all workers are coming in touch with the queen to ask her what they need to be doing at that particular age group. In contrast, what they're doing is picking up all of the pheromones that are released by the queen through contact, and maintaining colony or cohesion that way. So that's when we say that honeybees are very special, they are because they have reproductive division of labor, whereby the queen performs all the reproductive tasks in the female caste, and the workers do all the non-reproductive tasks. And a lot of that control is pheromonal. So chemical communication is key in honeybees and other social insects. So that queen will lay anywhere from 1,500 to even 3,000, some people have reported, eggs per day. And she can live anywhere from a year to 4 years. So imagine 365 times 4 times 3,000 eggs a day. So she is truly an egg-laying machine, while the workers are performing all the non-reproductive tasks. One of the tasks of workers, young workers, is to attend the queen. We call those queen attendants, or the queen's retinue. And the queen's retinue is a very important indicator, some of our research has shown, of queen attractiveness to workers, queen quality, and ultimately that queen quality will translate into increased colony growth and productivity. So if you were at any of the other talks that I gave, I showed a lot of data on how important that retinue of workers is to the queen, and how the retinue changes in size and activity level based on how attractive that queen is to those workers. So those queen pheromones will elicit several physiological and behavioral responses in workers, and that their biology has been such that the queens have come up with incredible mixtures of chemicals that are highly, highly attractive to workers. So the most important one, or the one that we talk about more, that it used to be called the queen's substance. Now most of the chemicals have been lumped, or at least the ones that have been synthesized artificially and used to probe worker attraction to these chemicals, are chemicals that are released by the queen's mandibular gland. So she has 2 glands, one on each side from her mandibles, that produce an array of chemicals, that more than 40 chemicals, a lot of which are still unidentified. But the more important ones are these ones that have really long names. They're really long molecules, organic molecules that have been shortened into the acronyms 9-HDA, 9-ODA, HOB, HVA, an enantiomer of this one, which means 2 different flavors of the same kind of molecule. So the 5 more important chemicals in QMP are these. And they cause, as I said, several behavioral changes. So how does that happen? Well, imagine that this diagram right here has a queen in the middle, and these dots in the middle are the queen's molecules that she's releasing from her glands, not just the mandibular glands, but the other glands. And so these are contact pheromones, and so the retinue workers are not only feeding and grooming the queen, but they're also picking up these molecules of QMP that in turn will change, as I said, the behavior and physiology of the colony. And so these workers will groom the queen, pick up their pheromone molecules. Then they will groom themselves, just like all bees do. They will try to put a lot of these molecules back into their forelegs and antennae. And while doing that and doing, you know, licking each other, and they will not just share those molecules with other bees around the retinue, so it's kind of like a chain effect that the retinue workers will pick up the molecules from the queen, then they turn, groom themselves, share it with other workers, and so on and so forth, but they also translocate or kind of internalize some of those molecules through grooming, and that also causes those physiological changes in the workers. So QMP modulates behavior, as I said, and this is just a non-exhaustive list of all the things that QMP does in a colony. And all of these short sentences that I am saying, like, you know, oh, it just elicits a retinue response. All of these things have been summarized from years and years of work by several researchers that have looked at QMP. But QMP elicits that retinue response right here. It inhibits worker ovary activation, and we'll go into that. It inhibits queen cell production, so queen rearing, at most of the year. Unless there's some emergency queen rearing or supersedure going on, the colony will maintain their one queen situation because of her QMP and other pheromones released. QMP is also known to stimulate pollen foraging and brood rearing in small, newly founded colonies. It increases nectar foraging, delays the age at which foraging occurs. It reduces the production of juvenile hormone, which is an important hormone for caste differentiation or age polyethism. And also it attracts drones during mating. So virgin queens have a very unique perfume, QMP perfume, that is deadly attractive to drones during mating. Very different in chemical composition to the composition of QMP in mated queens. So as I just said, QMP composition varies based on reproductive state, so whether they have been mated or they're still virgin, or on volume of seminal fluid that it's stored in the queen's sperm storing organ. I'll go into that. So it will depend on her reproductive state, on how well she mated, how many mates she mated with, the development of the queen, and so based on the age at which the larva, the fertilized larva, is chosen to be raised as a queen, for instance, during the grafting process, if you're in the queen rearing business. So I won't go into those data here, but other environmental factors and biological factors will affect QMP composition. QMP and queen release pheromones, as I said, have a very strong attraction to workers. And so a lot of studies have looked at, you take out at how attractive QMP is to workers. You can dissect out the queen mandibular glands. Even you can do it in a quick and dirty way as a beekeeper, just to see how attractive these gland produced pheromones are. You make washes out of those glands and then put them on filter paper or a Q-tip, and immediately you see worker attraction. So for instance, here in Petri dishes where you're doing these kinds of experiments, you just put a little swab of that extract in an arena, and you will get very, very high attraction of workers toward those washes of the queen mandibular glands. As I said earlier, QMP inhibits worker ovary development. So here in this diagram we have the typical look of a virgin queen's ovaries. Of course, she has two ovaries, one on either side of the abdomen, and each ovary contains these strings that contain all the eggs. They're called ovarials, and each one of those ovaries contains anywhere from 120 to 180 ovarials, and that also depends on queen quality. So if you see what the inside of a queen, either a virgin or a mated queen, is compared to workers, you will see that laying queens will have very developed ovaries compared to the ovaries of normal workers, which typically don't have any ovary activation, or of those laying workers that will activate a couple of ovarials with a few eggs that they use for laying unfertilized eggs. So in terms of size, there's no comparison between this is a laying worker, a virgin queen, and a fully mated queen. As I said earlier, it also QMP inhibits queen rearing, so typically the nest will have some cups, kind of just keep them there in case that there's any need for rearing any queens. So starter cells to rear queens. But in the presence of the queen pheromones, those remain unoccupied. When you are, when there's either no queen or a very crowded condition so that workers can't pass on that QMP, the colony will start rearing queens. Typically, as I said, in the periphery of comb because they need more space. So QMP inhibits worker ovary activation and queen rearing. So let's talk more about what's happening in these normal swarming conditions when we have a bunch of queens being reared, because all of those conditions are being met that I mentioned before. When you have several queens being reared for a swarming scenario, you can look at what's going on in your hive just by looking at the cells, at the queen cells, and what's happening in them. So if it's the swarming season, you might see either one of these cells or one of these cells. So this will indicate that your queen has properly emerged. She will chew her way out of the queen cell, sometimes leaving the little bottom flap still attached, or it will just completely come off. And so that will be a sign of proper queen emergence. So you'll likely have at least one or a couple of virgins that recently emerged that are kind of roaming around, and they're starting to start these competitions that I will mention about between each other to kind of fight until they can inherit the nest. If you see anything like this, where you have a hole on the side of a queen cell, no proper emergence at the bottom, and sometimes even a worker inside that queen cell, it might be that, well, it will be that the queen did not emerge properly. The workers either took her out themselves and then stayed in. Sometimes you hear stories of people that saw a worker inside a queen cell and they thought, oh my gosh, a worker was reared in a queen cell. It probably was that she was helping to get rid of the body of the deceased queen, and then they sealed her up, and there she is, the worker in that cell. But that will be an indication that that queen, or many more in your hive, did not emerge properly. But when they do, and you have several of these, and as I said, queens can be reared at kind of different times, and their emergence can be at the same time within a matter of hours, or it can be at very different even days, because the queen laid the eggs on different days in those queen cells. So the queens do not emerge synchronously. The first one to emerge usually is the one that will win a lot of these duels. We call them queen duels. So she will seek out developing queens and start stinging them to death. So queens, before they mate, they have a stinger, and they can sting each other in these competitions. And unlike workers, they don't have a very barbed stinger. And they're also stinging each other, and they're insects, so they have a very soft exoskeleton, so they can sting, and there's no barb in the stinger, so they can release the stinger without it remaining inside the queen, so they can sting multiple times during these competitions. If multiple queens emerge successfully, they will fight to the death, as I said earlier, and the winner of these combats typically is the one that inherits the nest and is ready to mate. Some studies by some of my previous supervisors have shown that you can put virgin queens in petri dishes or arenas and get them to fight with each other, and typically the one that wins is slightly larger in size and more vigorous. And interestingly, the workers leave it up to the queens to do most of this fighting. So they kind of just take a back seat and wait for them to fight it out. Why would that be? Why is that a good alternative versus themselves killing the virgin queens? But typically, yes, they will leave it because typically the strongest queen will be strong enough to kill her competition and still be healthy enough that she's not too injured to take on the egg-laying duties. So after emergence, here's a picture of a queen that's coming out. The queen that wins these duels will come out and roam around. Once she's kind of gotten rid of the competition, she will take some time to start spreading her QMP as a virgin. But at least they start learning what that new queen smells like. She also takes orientation flights, just like the other bees in the colony, to learn the local landmarks. And about 7 days after emergence, but it also depends on weather, she will start taking nuptial flights. And about 10 to 15 days after emergence, and only once properly mated, she will start laying eggs. So as I said, they take orientation flights. They take about 5 to 10 minutes each. They're short distance, and they take place near the hive so that they learn the local landmarks around their hive. Basically, they know what home looks like. In an apiary where all of your colonies are white, there are no differentiations between colonies, and you have a bunch of queens being mating around the same time, you might have situations where the queens misplace the beacon of their own home and they can go into someone else's home because they kind of miss the entrance to their home if they don't really look very different. So that's something to, it's always good to have some sort of recognizing sign of the entrance of each hive so that they can, both queens and drones can go back into their own colony instead of drifting to the one nearby. And then they take their mating flights, which can take anywhere from 5 minutes to more than 30 minutes or an hour. And they're usually very far away from their source colony, 1 to 3 miles away. So they can either take one mating flight or two, or even 3 or more. And they somehow, we still don't understand how, but physiologically, they make the decision that they are done mating and they don't go out. About half of queens take only one nuptial flight and they're done, and they will never mate again for the rest of their life. About the other half, 45% will take 2 mating flights, and only about 5% of queens will take more than 2 mating flights and then call it quits. And those can happen within the same day or within an extension of days in a time period. This is what the diagram of the queen's reproductive organs, and the ones that you kind of want to remember is, you know, this is the abdomen. So that's why the queen's abdomen is so large, because it has to encase all of these ovarials that have the thousands of eggs being produced. But the other part is really important. This organ is the spermatheca, which is the sperm storing organ. And then when copulation happens, the endophallus of the drone will come in through here, and the semen will have to pass through the vaginal, the valve fold, and accumulate here in this kind of pouch. And then it will take about 48 hours for the sperm from her matings to migrate up these viaducts to the spermatheca. This is a common picture that people use when they're doing any kind of queen rearing to show what the spermatheca of a virgin queen looks like compared to the spermatheca of a mated queen. If you're in the business of rearing queens at all, typically you will sacrifice a couple of your queens, and you can even do this in your mating yard to see if your queens have properly mated. You can dissect out the spermatheca, is the size of a pin end, and you can remove with your fingernails the tracheal net. There's a net of tracheae that provide the oxygen to the sperm inside the spermatheca so that they can stay alive. But if you have the spermatheca of a virgin queen, is basically just some saline solution, but no sperm inside, whereas the spermatozoa in the semen or the semen and the spermatozoa look kind of a marble brownish color. And so when you find a queen like this, you know that she was properly mated. At least she mated with some drones. We don't know the quality of that semen, and that's a lot of the stuff that we do in our lab, because it turns out that a lot of the sperm that is in the spermatheca is not viable in certain conditions. So not all of the spermatozoa in that spermatheca can actually fertilize an egg, but at least you have semen and some spermatozoa in there. So what about the other half of the coin, the drones? Drones take 24 days from egg to emergence. So again, 3 days as an egg. On day 4, they transition to a larva. On day 9, the larva is capped. And then on day 24, they emerge, plus or minus hours, again, as in every cast. About 12 days after emergence, the drone will become sexually mature. Some of the work that we have done in our lab and other collaborators have done, however, have indicated that that's a very low number, 12 days post-emergence. That sexual maturity in drones typically takes longer. So it can be anywhere from 15 to even 21 plus days before drones become sexually mature. And it might all have to do with environmental conditions and nutrition inside the hive to determine at which point the drones in a colony will become sexually mature on average. So there's a minimum of 36 days from egg to emergence between for drones of sexual maturity to become available, which is very important if you're in the business of hoping that your virgin queens will be properly mated, because they can be going out trying to mate, but there are no drones that are sexually mature in that area or wherever the queens are going. And so that might be one of the reasons why they're not mating with the proper number of drones. Interestingly, drones emerge with all the spermatozoa that they will ever possess. So they don't really undergo like a puberty per se in spermatogenesis after a certain time post-emergence. What happens with drones, they emerge with all the spermatozoa that they will ever have, but it takes those 12, 15, 20 days post-emergence for the sperm to migrate to the seminal vesicles before they can mate. So again, that time, sexual maturity is not for the creation of spermatozoa, it's for the migration of the spermatozoa to the seminal vesicles. The colony will produce thousands of drones only if the right conditions are met, if the colony has enough worker force, if they have the proper comb available for drone production, and if they have the resources available, enough food, especially brood food. And then young drones are fed inside in the middle of the brood nest. So one good indication, here's just a picture of a queen cell, a queen that is probably on her way out, coming out, and then sure enough, during the same time, you have the production of drones from those very much larger cells than those from worker brood. If you're looking for what indicators are good to assess sexual maturity in drones, for instance, young drones are hang around near the brood nest because they're typically being fed by the workers. When they reach sexual maturity, they like to go on the outer edges where the food frames are in a kept hive, or in a tree cavity. They will go to the edges where the food is being stored, and they congregate on the outer edges. And then what they do is they try to go and mate, and then come back and refuel with food, and then go back and try to mate, etc., etc. So just like the queens and the workers, they will take orientation flights, and then they start leaving the hive with more, they dare to go further and further away to look for queens. If you have nice weather and nice temperatures, not such a windy environment, not a lot of rain. For example, if you have a lot of rain or you've had a whole week of bad weather, you will typically find that basically most of the drone population of sexual maturity will have left all colonies in that window of nice weather, because they all like the same kind of environmental condition. Nice, mild, weather some patchy sunny sky for orientation. And so bad weather can delay the mating of both queens and drones. And in the fall, the adult drones that are still around will be kicked out by the workers. They will either stop feeding them and or actively push them out of the hive and starve them to death and then say, bye-bye, that's it. There's no more queens to mate with, therefore there's really not a whole lot of use for very expensive drones. So they die before winter. There's really no drone population over winter. So the lucky drone that gets to mate will see his demise very quickly, but the vast majority of drones actually never gets to mate and then get kicked out before in the fall. So very unfortunate kind of conditions for being a male honeybee, really. This is a diagram of the drone's reproductive organ, and there's a couple of structures that are interesting to learn about. Here we have the claspers. We'll have some picture of that in real, like a real picture of this. You have the claspers. They're the older or more sexually mature the drone is, the more orangey the end of the claspers is, which is quite interesting. We don't really know the exact function of that, but the claspers are of the color, but they are really good in holding on to the queen during mating. We'll go step by step to show this. There are very important hairs right before the bulb, and these hairs help in sexual competition. So the hairs are hypothesized to help remove some of the sperm from the previous mate, and then the drone will insert his endophallus, endo because it's outside, and the bulb of the endophallus is what goes into the queen, because over here you will have a pool of mucus, and then on top of that the pool of semen that will go into the queen. So here's a picture of those claspers, and they're used during mating to hold on to the queen. Here you have that pool of mucus. It's kind of white, very thick. It provides a lot of proteins to the semen, and then on top of that you have that marble color semen that I showed a picture of earlier. Mature drones have about 10 million spermatozoa in their seminal vesicles, but only about 1 microliter, tiny millimeter cube of semen. And it even gets even more diluted because a drone only provides about 500,000 spermatozoa to a queen per mating. And if you can think of the spermatheca as an organ that can withhold or store about 8 to 10 microliters or millimeter cube of semen, then you can start doing the math that to fill it up, if only a drone will provide about half of a microliter, then you will have to mate with multiple males so that you can fill up the spermatheca that can hold 10 microliters of semen. So and that filled spermatheca with about 10 microliters or so can contain anywhere from 3 to 5 million spermatozoa. So just think about mathematics, 3 to 5 million spermatozoa, because she needs to fertilize how many eggs per day if she's laying worker destined eggs anyway. So this is what people will do to artificially inseminate queens, which is a feat just in itself. It's really expensive to do, and also very tedious, and it takes a lot of practice. But they will take up tiny syringes that will collect that semen from sexually mature drones, and then either mix them up or decide what to do with that semen to then push that semen into the reproductive organ of the female. It has to go through that valve fold and go inside. And then it will migrate up to the spermatheca about 48 hours later. So now that we have the A, now we have the B, let's do the A plus B and do the mating, how mating occurs and what DCAs are. So that virgin queen that finally is able to go out and exit the hive to mate with drones will exit, as I said earlier, when the right conditions are met. Temperature has to be above maybe 20 degrees Celsius. It has to be sunny with some blue sky patches because, as you know, honeybees use polarized light for orientation, and so they want some patch of blue sky to know exactly what time of the day it is and to help them orient toward home. And little wind. So when it's really windy, it's dangerous because they can be blown off and get really basically lost, which happens. You can have queen losses during mating. It can be dangerous out there for these queens. They can either get eaten by a bird or they can get lost during windy or rainy days. So then she will leave the hive and fly to what are known as DCAs, or drone congregation areas, where hundreds or thousands of drones await for a time to mate. There are typical pictures of people that have tried to locate DCAs. There are people in the United States, probably here in the UK too, that they're known as the DCA whisperers. And they know where DCAs will form, and then you can take either a virgin queen and tether her to a helium balloon, or you can take a queen lure and then start looking for drones, because they, again, that QMP, amongst other things, it's highly attractive to drones, and so the drones will just follow the smell and then visually locate that queen and hope that they will be able to mate with her. So DCAs are very difficult to locate, but people that have this ability to locate them have, they follow specific landmark contours, areas where there's some landmarks, but also areas of open space so that mating can be happening without getting into the way of very large trees or buildings, etc. So there are no general rules about where DCAs are located, but they are temporally and spatially quite consistent. They will happen around the same time, around the same area every year. So once you've discovered the DCA, you're very likely to find it the following year. So queens don't usually mate with drones from the same apiary, and that's something very important to learn. Drones, to avoid inbreeding, the honeybee has developed amongst all the wonderful biological strategies. One of them is that queens will fly far to mate, whereas drones will stay relatively close to their source colony for mating. And so that separation of queens going far and drones coming later will avoid inbreeding between queens and drones. But here, for example, in this picture, you have the location of apiaries here in the squares in one area, and then you have the dots where DCAs are located. So all the dots are DCAs. And then you have another area where DCAs are really far away from the apiary, and so you really don't know the general rules of where DCAs happen. It's more like geographic contours. Drones emerge earlier in the year and stay out longer in the year for mating compared to females or queens, which are more constrained seasonally as far as when they come out to mate. And they also come out earlier in the day and stay out later in the day than queens. So queens are more restrictive too during the day. What time of day? So you can see that most mating will occur between 2 or 3 p.m. and 5 p.m. when it's a nice, sunny, warm afternoon. Once that queen finally goes out to mate and one lucky drone gets a hold of the queen, this is what happens. The drone mounts the queen, uses that clasper to hold on while inserting the endophallus. So finally he's able to release the endophallus. It will never go back in. Very difficult to put all of that structure inside. And as you will see, he can't because he will die after this. So he inserts the endophallus, then he lets go of the queen, but the bulb is already in, and then the bulb gets ruptured, and the drone then will go and sadly die. A happy drone. And inside the queen will have remained the bulb with the semen of that drone. This is kind of the A plus B together. You have the queen's reproductive tract, and you have the endophallus of the drone, it will have gone in past the valve fold and the spermatozoa remain inside that pouch. And then, as I said earlier, it will take about 2 days for those spermatozoa to migrate up to the spermatheca. So when the drone comes out, drops to his death, and stays with, keeps the bulb inside, then the next mate, amongst other things, will take out the bulb from the previous mate, use those hairs to remove some of that semen from the previous mate, and then insert his own bulb into the queen, and so on and so forth, until she says, I'm done. I'm satisfied right now. Let me just go home because I'm hungry. And then she goes back in, and then the workers are the ones that remove the last bulb from her last mate, either for the rest of her life or maybe for that nuptial flight. And so she will end up with what we know in beekeeping as the mating sign that you can see from queens that have just returned from a nuptial flight. Honeybees have multiple mating, as I said earlier, and a lot of the research has shown that Apis mellifera, depending on the strain, mates with about 13 to 14 mates. So my previous advisor and his collaborators did a lot of work on looking at the mating frequency, or how many partners the queen has mated with. And you can see queens that are not very well mated, that have only mated with 4 to 7 drones. Then some, the majority, will have mated with about 12, 13, 14. Then you have these outlier queens, very promiscuous ladies that will indulge in multiple mating more than the average amount, but typically you have 15 or so drone mates. And that genetic diversity has been shown by several people to confer a colony with several different genetic lines to combat disease or increase productivity or specificity in any kind of foraging activity. So very important to keep that genetic diversity in the hive. And by the way, polyandry, which is multiple mating by the queen, is really not very typical of in the social insect world. It's quite unique for honeybees to be so polyandrous. So what are the take-home messages for this basic mating biology class? Understanding the mating biology of honeybees will help all beekeepers, beginners and advanced, to remember, regardless of expertise, how bees are preparing for the mating season so they don't get any surprises. So there will not be surprises in the queen rearing or swarming, because you know exactly what's going on, how many days it takes for this to happen, at least the minimum. And understand that natural insect of mating, swarming, and dividing. And also, if you're in the business of providing proper mating for queens, to be conscientious about the other side of the coin, the drone biology, because up until recently, people were not really paying too much attention to drones, but they are providing half of the genetic material for each worker in the colony. And if we don't have enough drone sources, we don't have sufficient numbers of mates for our queens. So we'll be talking more about the mechanisms that initiate swarming and how swarming is decided upon by a colony tomorrow, and that will show a lot more like scientific findings. But for now, I want to encourage you to visit our Facebook page from our lab. These are my 3 graduate students. Liz works with queen reproductive biology, so trying to understand the factors that affect reproductive biology. Adrian works with drones, so the other side of the coin, and what factors influence drone reproductive biology. Pierre is working on projects with pollen in urban environments, so I wish I had time to talk about his project because it's really interesting. But we always post the newest and most interesting research that comes out, not just out of our lab, but labs in the United States and internationally. We also have a website, but it's not as updated as the Facebook page, honeybeelab.tamu.edu. But I would like to end with a huge thank you to the sponsors, of course, Manlake UK, the British beekeepers, especially Simon Crowson for inviting me Texas A&M University for funding and allowing me to be here today. And also be talking about our project with feral honeybees that we've been doing in New Mexico and Texas with Africanized honeybees later today. So that will be fun. But I don't know if we have time for questions, but I really thank you for your attention.
Do you think the practice of drone removal for varroa control compromises queen breeding?
Does the removal of drone comb for the purposes of varroa control hinder queen mating? And the answer is, if it's being done during the time where queens are mating, it probably will. Because there are thousands of drones. If you have a full frame full of drones on either side, there will be a couple thousand drones at least that will be produced by that frame. So you have to be really conscientious of your drone availability and also that of your neighbors, especially in a country that is smaller and has more beekeepers nearby. You can probably assess roughly if there's going to be drones available for mating.
If I may ask, why laying worker lay few eggs into one cell?
More than one egg?
Yes, because laying workers lay more than one egg to the
They're
cell.
not really wired for egg laying, and so it's not really a practice that they do, and so it's completely kind of a weird wiring, and they will just drop them in. Typically, if you see in a normal colony, other workers will police each other's egg layings and they will remove those eggs. But when this happens, they're just going everyone for themselves. Let's just lay as many eggs as we can. We don't know where it is. They're not really laid neatly like a queen laid egg. So we don't really know why, but it's just a weird, unnatural kind of wiring crossing that's going on in workers.
You implied in your talk that some drones had inferior sperm. Is this a natural phenomenon or?
Yes. What we know thus far is that all drones will have spermatozoa that are not viable. Therefore, they cannot penetrate the cell wall of an egg and they cannot fertilize an egg. They're basically dead sperm. But very little has been done with honeybee sperm as far as motility and vigor or other aspects of fertility that are studied with like mammals and human fertility and sperm. But we know that viability, that is whether the acrosome and the cell wall of the head of the sperm is viable or not, is something that we have found to be present across queens. So typically queens will have, if they're good queens with good sperm, they can have anywhere from 80 to 95% sperm viability, whereas some of the ones that are subpar, they can have only maybe 40 to 60% viability. That means that 40 to 60% of the sperm is dead. It will not be able to fertilize an egg.
Is it known how the queen finds the DCAs?
Is it known how the queen finds the DCAs? Not to my knowledge. It's visual. There's thousands of drones. So instinctually, they will just fly where the thousands of drones are. Someone is right now studying drone-released pheromones or scents that could potentially be picked up by queens, but that's very new work that we're not doing. Some people at Penn State are doing. That's curious. And visually, of course, they will locate them, but we don't know if they're releasing any kind of chemical that can help them, just like it helps drones find virgin queens. It's a good question.
We've been doing our splits over the last couple of years. We've noticed quite a marked increase in percentages of matings when we've made the whole yard queenless as to doing splits and leaving some queens behind, more or less. Have you had better strikes when you make the whole yard queenless, or compared to doing splits when you're leaving the old queen behind?
We typically don't do that practice of leaving a whole yard queenless. When we're splitting, we typically split when we have already purchased queens somewhere else.
Putting cells in.
Or cells, yes.
So you've never split the whole queen, made the whole yards queenless?
Quite honestly, we can't afford to see if the colony will just raise their own queens, because we use the bees that we have in our apiary are for research. So typically we're either very controlled in what kind of queens we want in that experiment, for instance, or we're limited in the number of bees that we have, and we just won't leave it to chance. For example, this year was the worst year that I've seen as a beekeeper for the queen rearing and mating scenario. And it's not just us, all the queen producers in the area where I am in Texas suffered the same problems, and we are blaming the weather. We had very unusual weather this year, very wet, very cold spring, I mean for Texas, but still cold, wet spring that would just not allow the queens to go out and mate. The same for drones. If they mated, they were not properly mated. They would get replaced right away or killed by the workers because they were just not properly mated, and there were no drones produced in the numbers that we have. So we couldn't really afford to just allow a colony to requeen itself. I know that people do it, especially if you're a small-scale beekeeper. Natural requeening is fine, but...
Cells in.
cells.
We're putting cells in.
Yeah, we put cells in too.
You make the whole yard queenless and then requeen the whole lot. And we've had a far better strike of doing the split and leaving the old queen in.
Wow, that's good. And it's cheaper if you're just using cells instead of queens, right? Typically?
In New Zealand, I think we're all cell operation queens.
Yeah. Oh, okay. Cool.
Right. Thank you very much.
Thank you again.
Thank you
We'll
very
see you in the afternoon.